Hello, everyone, and welcome to today's edition of GIFS Presents Engineering Biology Speaker Series. Thank you all for joining us this afternoon, this morning, or this evening, wherever you are around the world. Welcome. My name is Steve Webb, and I'm the Chief Executive Officer of the Global Institute for Food Security, or GIFS, at the University of Saskatchewan here in Canada. Bit of background on GIFS. We're a public-private partnership between Nutrien, the province of Saskatchewan, and the University of Saskatchewan, and we were established in 2012. Our mission is to work with partners to discover, develop, and deliver innovative solutions for the production of globally sustainable food. And sustainability has three important legs: economic, environmental, and social. And we serve as Ag's innovation catalyst. We connect up and down through the ecosystem, and we advance innovations to bridge the gap to commercialization to deliver resilient and sustainable food security for all stakeholders. The inception for this webinar series that we've been conducting was actually inspired by Dr. Angela Bernard-Hahn, from the Dean of USask College of Agriculture and Bioresources. Angela had asked if I was going to give a seminar to the faculty and students at the university on engineering biology, and we decided that it would be actually better if we brought experts in from diverse backgrounds and experiences with the technology to actually provide a webinar series. So we took Angela's idea, and hopefully through this series of 6 events that we'll be able to show the breadth, depth, and impact that synthetic biology or engineering biology will have, not only on ag and food, but in the context of several business verticals right across the entire economic ecosystem. Our first edition was a couple weeks ago, where Dr. Patrick Westphal, the Director of Cell Biology at Inscripta in Boulder, Colorado, gave a great overview and an introduction to the field and provided examples of how it can be used in ag, health, and medicine. And I want to let everyone know that his presentation, a video of his presentation, is available on the event page, and you can watch it. And again, I think Patrick did a great job to kick off this seminar series. And today we're proud to have, or privileged and proud to have Dr. Steve Evans, founder of Renovate and a retired fellow from Dow AgroSciences, to speak about biomaterials, food, fiber, and feed, regulatory environment, and global scale. And a little bit on Steve is he's actually the guy that's actually accountable for us at GIFS pursuing the development of our engineering biology platform. Steve was a former boss of mine. I worked for Steve when I first moved down to the US in 2000. And Steve and I worked together for a number of years. And Steve has been very active in the engineering biology field through, at the beginning, Sinberg, EBRC, as well as other opportunities in the engineering biology space. Steve has been subpoenaed to testify in front of Congress about the impact that this technology can have, and he's been very active in the entire space as it spans both drugs, biomaterials, and agricultural applications. And again, Steve took probably 2 years to get it through my head about how this could be really disruptive and make a difference in how we do not only do research, which were some of the examples that Patrick used last week, but how the combination of automation, miniaturization with biology and engineering thinking coupled with computational power allows us to drive and accelerate the design, build, test, learn cycle. So without any more delays, I'm going to hand it over to Dr. Steve Evans. Steve, the floor is yours.
All right, thank you, Steve. Let me push buttons and get this going. Is everyone able to see my screen?
Yes.
Excellent. All right. Well, Steve, thank you for that kind introduction. And I think we've spent quite a bit of time together. It's been a fun ride, and I want to thank GIFS and the team that you've assembled for helping me have an opportunity to speak here. So as Steve did indicate, I'm retired now from Dow AgriSciences. I've spent a number of years working in environmental and agricultural biotechnology from the days of the mid-1980s, and had the privilege to be able to bring the first dead recombinant organism through the Environmental Protection Agency for an agricultural purpose. And so I reached back for a period of time in history, and I think that's what I want to try to capture in this particular talk. So the title, if you think about it, is quite daunting. These 4 areas are in fact ways in which you could write narratives of civilization itself. How we handled these things have both advanced areas, they've caused catastrophic disease areas when something failed, and we've been to war over some of these things. If you think about the subtitle topics, the regulatory, environment, global scale, these areas have really solidified their impact over the last 50 years. And so while I can't do a deep dive into these, what I'm going to try to do is use a series of vignettes to take us through these, and the slides will be available afterwards. The point that I would like to try to pull as a theme here is that sometimes as technically minded individuals, science and technologists and engineers, we keep ourselves removed from this interaction with society. Society here is not smaller. It's just tempting for us to view it as something that's distant, out in the future, will be handled by somebody else. And so part of what I would like to do today, I've used this word mashup to indicate science, technology, and society should be thought of as very intertwined, and in the past 50 years or so as having a concurrent development. So we are looking at global concepts around these materials. In agriculture, the term that is used quite frequently now is the 4th Industrial Revolution. Steve actually hit some components that will tie to our series today. So in looking at food systems, for instance, next generation biotechnology is a key component. It's going to be able to help us make new materials and new technologies. But some of the things that Steve just hit that Patrick talked about are also components that help define this 4th IR, 4th Industrial Revolution. This cartoon from a recent publication shows how we are attempting to move the dial here. So moving from an agriculture-based framework for producing the materials that we're used to consuming and other materials into an area where synthetic biology or engineering biology is enabling alternate production methods. So they'll come alongside for a while, and for some people, there's a desire that they would supplant them. So in thinking about this 4th IR, I think it's important for us to look backwards and think about what came before, which are these 3 prior industrial revolutions that took place on long time frames. If you reach all the way back, you can see something like mechanization impacting the way a textile could be both farmed and then brought to a new commercial reality. If you look at the 2nd industrial revolution, really what it did was help push mass production. And not only with the rise of petroleum, we had the real rise of synthetic chemistry. And so we began to be able to synthesize molecules. Fermentation took on a new meaning over what it had been doing for centuries. And then notably production of polymers such as nylon took place. When you look at the 3rd IR, this is a really pivotal time. It's traditionally dated to around 1969 and is known as the computer or internet phase. But it's also a period of time in which globally environmental policies and regulation took place, for us, it's also very near the initiation of the recombinant DNA story. And so one of the things that I think that's important is within 2 years of this date, within 2 years of 1969, both the US and Canada had established their respective environmental protection agencies, and groups like Friends of the Earth were also come into existence. And so when you start thinking about how fast may things move from lab to policy, many of you are familiar with this paper, the chemical synthesis of the Mycoplasma genitalium genome. This happened in 2008. By 2010, this particular publication had been the real crystallizing event that brought this before the UN Convention on Biodiversity. And so since 2010, this convention has been trying to grapple with whether synthetic biology is a new and emerging issue. When we start looking at how this has played out, this is an excellent review from last year from 2 industrial scientists. What I will leave you with is 2 statements they observe. This decade-long exploration into this new and emerging issues question, they argue is sort of like an exceptionally prolonged Asilomar conference. But what's very important, and what will lead into this next slide, is that the scientists and engineers that are practicing the technology are largely absent as practitioners in the discussion. One of the ways this shows up from a group of academic scientists in 2019 was in this publication in Trends in Biotechnology, in which they argue that we need scientists, policymakers, stakeholders to get actively engaged, because they view that the deliberations are going to change not only how products are made, but how research should be conducted in the engineering biology field. So this is one of the reasons I like to put this these 3 words together, because science, technology, and society need to be interacting together in a way that may not be comfortable or traditional. We all are familiar with the role of advanced genome editing techniques. And so this is a very nice review going country by country through what they can do to surmise and summarize the various jurisdictional readings on how genome edited plants will move forward. And then if you think about, again, a global aspect, the Sustainable Development Goals, the SDGs, are also ways in which synthetic biology is very much perceived to have an impact. In this particular publication from 2017, after listing several other areas, again, tying to our topic, what the author said was, last but not least, it can have a role in material production, if they can figure out how to decide to regulate it. As they move through some of these concepts on regulation, the very first thing they pull out is entitled this route to from laboratory to market. I call this crossing that lab threshold. And basically what they say is that synthetic biology things are a lot like a path for new drugs, but not really. They're more complicated. And I will show some of the differences in a couple of slides. And so as we pull up from this first vignette on regulatory, what I hope you can see is that there are significant efforts that have been placed globally. There are a number of regulatory and policy actions that are going on. Synthetic or engineering biology really received its own spotlight beginning in about 2008 in this arena. There's still a strong emphasis on this lab to environment threshold, and we're about 50 years into the recombinant DNA technology revolution, and yet early stage scientists remain erratically engaged in the policy and regulatory forums. And so once again, this framework that I'm trying to present. Now, I borrowed this slide excerpt from the United States FDA because they have a very long time frame of looking at how products and technologies have arisen during this rDNA revolution. And the beginning of this is clearly at they are within the framework of the third IR. And so what I'm going to do here is very quickly highlight is that at the beginning, where we were first looking at restriction enzymes and interspecies cloning, within about 10 years from that, we had a pharmaceutical product on the market, insulin. It took over 20 years to get the first agricultural product into the market, a GM tomato. Using that same time frame, it took over 40 years to get the first transgenic salmon into the environmental or into the approval phase. And then as the last they have on this chart is in 2019, a consultation was finished on high oleic soybean oil that had been genome edited. So we transitioned from the third IR to the fourth IR in this particular phase. And I'm going to hit a couple of these points as we move forward. So as we look at that inception of recombinant DNA technology, I've illustrated some things that happened in a very short time frame. This is one of the Asilomar conferences with some Nobel laureates in it. This is Cohen and Bowen, who went on to form the company Genentech, that was then largely responsible in partnership with Lilly for developing this first drug, the humanized insulin. So we went through a very short period of time where work that was really Nobel Prize level material in some aspects also spawned not only companies, but it spawned the formation of regulations very early on with the NIH in the United States. Something again that's important to note is that the Humulin went through the FDA in about 5 months rather than about a customary time of about 2 years. And so at that point in the mid-90s or the mid-80s, insulin had gotten out the door, and we were then ready to move a number of GM organisms out into the environment, this laboratory to environment threshold. This is a report that summarized a number of those, both microbes, fungi, and then larger animals. But something happened that basically put a stop to this. And I've picked this component to show 3 different lenses of the ice minus bacterial release that happened in the time frame of 1985 to 1987. And the 3 lenses are, this is how it was viewed within the academy. This is how it morphed when it was presented in science. And then this is how it was shown to the population at large in the New York Times. And so you can see the imagery changes dramatically. And by the time we get to this point, it was all about the hazard. And so this narrative, in essence, killed the movement of recombinant microbes into the environment for a couple of decades following. Now, the recombinant or transgenic tomatoes did continue to move forward. It took about 10 years after insulin. These are some pictures of those products as they were sold in the United Kingdom, and a little over almost 2 million cans of these flavor saver and other derivative tomatoes were actually sold. Something like the transgenic salmon took a quarter century to get from this developmental phase. It had begun in that mid-90 point also, became an official project in the late 1980s, but it took 25 years to get through both the Canada and United States official approvals, and then another 5 years or so for it to move forward into the market. 2021 has shown some interesting approvals elsewhere on the globe, though. After over 20 years, the Philippines did approve the so-called golden rice or GMO rice. And interestingly, Nigeria has been able to approve their BT containing GMO cowpea, which is going to be a very important component for the diet there. And then just to kind of relax back, if you're interested in a movie that blends biotechnology, theft of corn off computer files, and then feeding the world, the movie Unknown with Liam Neeson is a movie that is based on that. So these concepts do move in and out, not only the technology, but this fascination with the technology in our society at large. It's now 2021. At the middle of last year, Chris Voigt published a paper in which he tried to highlight some engineering biology applications that would change the world within a decade. We're all familiar with the Impossible Burger. That was the first of the agricultural or food areas that was highlighted. This is a new film called Highline that's produced by Zymergen. And then this is the high oleic soy from Calyx that was mentioned on the FDA slide. And then this is the proven nitrogen fertilizer that is being developed by Pivot Bio. And then a couple of biomedical applications. So engineering biology is clearly moving in areas of food and in areas of molecule and polymer production that I think are important. What I want to do, though, is step back for a moment and then notice that there are other global activities in play. This one is looking at insect proteins in both food and feed applications. And the copywriters at Bloomberg had a little bit of fun here, pointing out that it is now being moved through the regulatory system to allow pigs and chickens to eat bugs. The reality of thinking about this, though, is that there's already nearly $1 billion market. 130 countries around the globe, 2 billion people already consume insects as part of the food chain, but it's not common in North America and Europe. Globally, again, the FAO has published a report showing the importance of this. And what the authors of this publication indicated was that since that report, they've been able to see over 15,000 publications. Moving online. One of the components that is driving this is the use of the black soldier fly. It's a favorite for the feed arena. Within the regulatory space, the EU, US, Canada, New Zealand are approaching these as novel foods. But again, the Asian cultures are trying to work through formalizing something that they've been doing for centuries. And this is an illustration in Singapore where the society is being asked to engage concurrently in the development of this technology. And you can bring a box of your food waste and enable it to be used in the production of black soldier fly feed. A discussion around food applications wouldn't be complete without mentioning synthetic biology vanillin. This came to the forefront in about 2015. It was, again, a large component of discussions at the United Nations. But what Friends of the Earth have been able to do is in a very focused way, produce or develop a narrative that this vanillin is also a villain, in large part due to the way it may impact smallholder farms in the production of natural vanilla. And some of the challenges around that have then led forth that some of the value chain producers in foods have concluded that synthetic biology or materials derived from it may be inconsistent with their company positions. So I'm going to pull out of food for the moment and jump into fibers and materials so that we can keep moving. The idea that we have industrials or textiles as being part of the fourth industrial revolution is consistent here with the European document. This report on plastics is framed out of an activity at the UN also. And so one of the things that I would like to do is spend just a moment and look at the history of plastics, because those were originally something that was made from biology. So you would take a biomaterial, do some chemistry on it, and a plastic would be formed. This dates back to the late 1960s, or 1860s, where there was an effort to replace ivory with a man-made material. Who hasn't made plastic from milk as a science fair or a science activity? But this was a commercial endeavor in the early 1900s, and that was one of the primary sources of buttons at the turn of that century. Soybean-based plastics were moving along very extensively in the 1940s. They play a minor role at the end of It's a Wonderful Life, again, societal fascination. But what changed significantly was the development by DuPont of synthetic polymers, nylon, in the mid-1930s. And again, this is about 70 years after this first attempt to begin using man-made items to replace biological components. So this one did eliminate the biology component. These things were so intriguing that apparently at the time, they were so-called nylon riots. This is a picture of the car that Henry Ford had developed. It had 14 panels in it that were made out of a plastic derived in some manner from soybean. And at that point in time, it took 1,000 pounds off the weight of the car. In the movie Sabrina, the plastic that was going to be made from sugar was one of the key get-rich ideas there. And it's kind of funny. It was going to be transparent, but they decided to call it bulletproof and scorch resistant. It was going to be natural and also edible. And so once again, the idea that science could do something intriguing has a strong component and fascination with our culture at large. And what brought a lot of this to stop was World War II, because what the Allied groups needed to do is replace rubber and silk with alternative synthetic materials. At that point in time, there were abundant petroleum supplies. And so there was a narrative that was developed that the use of petroleum-based products was in fact a conservation activity to conserve these other natural resources. Now I'm going to dive in for just a moment and look at one of these. This is polylactic acid. I've got two components. Polylactic acids and bioplastics per se started a resurgence around the year 2000. Again, this is about 70 years after the advent of nylon. Currently, PLA has about 300,000 tons per year that's produced. About half of it is from a single company. PLA is finding a lot of new uses. You can order it from Amazon for your 3D printer. There are some recyclable coffee capsules that are being made from it, and it's also being used as a new component for N95 medical masks. Production is happening globally and is increasing globally. And net, if you roll all of the types of biopolymers up, they contribute about 3.8 million tons per year to the plastic market, but that's only about 1%. And so this second part of it is something that is where the interaction with society, again, is important. There is a conflation between biodegradable and bio-based. With PLA, there are certain conditions in which it is biodegradable, but not all bioplastics are. Right now, again, they contribute about 1% to the total plastics market, they do so with just a fraction of the world's arable land. The challenge is that if you want to increase this number, you may need to increase this number. And then the biochemist at the university or at Michigan State has argued that to change that number markedly, you may have to increase corn production in the US to the equivalent of 20 to 25 United States. Plastics Action Center in Canada is one of the areas, again, that interacts through this ubiquitous infographic. And so you can go take a look at some of these facts within that group. And because of this, what's interesting is the biopolymer market is moving to things that are significantly upstream and are moving into classical chemical areas. So this is an example with Braskem and green ethylene. Now, what's important to note here is, again, the normal route for ethylene production starts with crude oil. What is becoming increasingly economically practical is to start with biomass and then make ethanol and then dehydrate the ethanol to ethylene. The important thing that if you think about this, while this is biology, the back end of your chemical plant or your ethylene production plant is going to look very much like a traditional chemical factory. And the way this then comes into play in thinking about biology, this is where I'm going to move to the end of the talk. This is a new document from the biofabricate area, and they're an industry consortium in for the fabric arena. And what they have on their cover is, again, this confluence. This is a parka made by North Face. It uses a material made by fibre, which is an biosynthetic yeast or a biosynthetic silk structure. And then they're showing pictures on the front of this fashion magazine of an SEM. Part of what they're having to do, though, as they bring this into their use community, is develop and explain this taxonomy around biomaterials. This is the version that they have in the Venn diagram, starting with biomaterials, going through bio-based, biofabricated. It's not important that we triage this now. It's just important for us to think about what this means. So this is a group of consumers, in the sense they're in the fashion industry using these materials. And so they are trying to be able to communicate to their industry group, but then also to consumers about what bio may mean. And so they start out with this term biomaterial being something of really a non-specific biological association. It can mean lots of things. And so they try to then work through a taxonomy of where biology is. Is it making it? Is it making a component that's inactivated, that's activated by chemistry, and so forth? And so what I'm going to leave us with then, my last slide, is what I would call this challenge. So engineering biology is absolutely poised to impact things in this topic area: materials, food, feed. It is not without headwinds, though, and it's not without alternatives that will compete in the marketplace and in the regulatory world for attention. Environmental applications still take much longer on average than the biomedical applications. And there are multiple examples where this is 20 years or more to get something into an environmental application. Some things with the plastics, they're new now in a sense, but they were also new several decades, sometimes 7 decades ago. And so it's important to be able to go back and figure out where things were in that point in time, and what both enabled them and what then might have caused them to be shifted away from. Overall, bio is not a clear message for consumers, and for regulatory people in addition. And so what I think that means for us is that we really need to take this concurrent development of science and technology along with the societies in which they're going to operate, and we need to act on that. We think it is probably more crucial now than ever if we want to maintain momentum and accelerate the role that engineering biology can play in these large challenges that are set before us. And with that, I will stop sharing my screen and stop.
Steve, thank you. Wonder why my camera, there we go. There we go. Steve, thanks for that talk. And again, I think it's the message about the opportunity for engineering biology, the breadth and depth of its potential to disrupt was clear, but also the importance of working together and collaborating and building a regulatory framework. We've seen where that was not done as a barrier to being able to bring innovations and fully benefit from the power of innovations with related to transgenic crops. Again, they have been proven to be, well, they've been around in the market for 25 years. I remember I saw my first herbicide tolerant canola crops 25 years ago near Laird, Saskatchewan. And I remember looking at the plots and saying, oh, wow, that's the future. That is the future for agriculture. And unfortunately, those technologies entered an environment where the regulatory path was not well defined. And there had not been the building of consensus and engagement with society and everyone to be able to bring those along. And it's probably one of the biggest tragedies that has fallen on the planet, because where those crops are being used, we've seen environmental benefit, reduced pesticide use, the ability. Stuart Smythe from the University of Saskatchewan has got along with others, have got a great series of papers out there of how they really made no-till work in the context of Western Canada, which has in essence changed how agriculture here is part of the solution to climate change, and we're a carbon sink. And I think it's a cautionary tale for us when we think about engineering biology and synthetic biology. And even those names are not the best names for the tool itself in the context of perception from the general public. At GIFS, we believe in that word sustainability. For us, I said, had three important legs: economic, environmental, and social, because we need to have that engagement early on. Our area in the food and agricultural technology space is fraught with challenges of being able to bring these technologies forward because of consumer acceptance, end user acceptance, and they happen very, very early. I liked your first slide about society isn't a long way away. It's actually right here, right now. So again, I'm talking too much. I like the slides. I like the presentation. And I was hoping when you talked about the ice story, you'd talk about where Friends of the Earth were positive on the technology as an example of how things have changed in the context of perceptions from various stakeholders across the ecosystem. For questions from the audience, please use the Q&A section. And Steve, we're gonna go to a question from a former colleague and friend of ours, Sequin Wang from Corteva. His question relates to Bob Langer at MIT. had coined biomaterials for materials to be used in medical applications. Do you agree with that definition? How would you put that into the Venn diagram?
I think the thing that we need to keep in mind is that user domains are going to define their terms and in terms that make sense. One of the things that is true across, say, take fashion and medical, materials and textiles are critical to both of them, but they may have very different functions. And in some cases, they may have different regulatory frameworks around them. So I think that sticking bio on the front of almost any technology or category is common. I think that it's up to then the communities to be aggressive in managing their own definitions. And that's what I'd say that we're seeing maybe as an issue on the back end with the materials going into the fashion industry, is that bio has begun to mean too many things to too many people. I think then it's also important to think about identify and work with areas where there may be some conflation going on. So that example being that biomaterials are always more I'm having a brain lock, that the materials are biodegradable. And just identify those areas and then work with both the society at large and with people who would produce and consume those to make sure that mixed messages don't get out. And so I think it's just very much about thinking through the life cycle of materials and thinking through where they're going to go and how they are going to interface with consumers, society, and regulators.
Excellent. And again, I think that the circular economy and even the polylactic acid-based bioplastics, they do decompose under compost conditions, but under cold temperatures, if they stay. So again, thinking about that, I think is super important. Another question from the audience is one that is near and dear to your heart, Steve. And also, I think one that is a call to action for scientists to engage in the process, to be part of the discussion group like you highlighted during the course of your talk. What do you believe needs to happen for environmental applications, the release of synthetically biology-derived organisms or engineering biology-derived microorganisms to be processed quickly? And what can people in the field do better to
I think that
context?
Yeah, I think that's a great question. I was involved in a National Academy study a couple of years ago called The Future Products of Biotechnology. And so that report that we published actually put together a framework for the US within the US framework, right? All of these have to take some account for the jurisdictional components that are in play. But the logic that we used in that report was to say, when you are looking at the first of a kind application, you should obviously spend more time and have more diligence and activity applied to that. When you're looking at the fifth version of something going out, it's not that you just make this a postcard, but you should balance your resources based on the novelty of what the application is. And so that is an area that I think has some logical traction and that one can begin to build on the experiences and knowledge that you have from prior regulatory approvals. I think that what the GM arena had to deal with early was the so-called the intergenic movement of nucleic acids, and that had questions and concerns. Not all of the technologies that are in place right now that may move to the environment are intergenic. And so that means they're going to fall into a different regulatory classification. And then I just think that it's important for, I would say, the overall life cycle of a material or a product to be taken into consideration, looking at the risks and benefits overall, because there's a risk or a benefit to not doing something also. Now, the challenge is some regulatory frameworks don't allow risk-benefit analysis, but I think it would be important in areas that do to utilize that.
You know what? Just a quick follow-up about the risk benefit. And again, there is risk benefits with, like you said, doing something or not doing something. And sometimes not doing something is more risky than actually doing it, and vice versa. What's your context on process versus product? Because when you think about the, for example, Humulin, the humanized insulin, it was functionally better than what was in the marketplace before, animal derived, but it was essentially an existing product in the mold. Why did that go so fast versus, or is that not a good example of product versus process?
So what I can't say, because I didn't go do the details on it, and at that point in time, I was ag focused, less so biomedical focused. So I don't know why it did move quickly. I think it would be interesting to see. Technically, so the NIH guidelines that were mentioned on my slide came about, those were 1976. The coordinated framework, which is the way that the United States chose to do the larger regulatory analysis, that coordinated framework came a little bit later. And basically what the coordinated framework says is we already had a number of large regulatory bodies, the EPA, the FDA, the USDA, and then a few others. Net, it said, did we need an entirely new way to look at something because of this new process, or were those agencies both statutorily and technologically equipped to handle any products that may come through? And the decision for the US was that the agencies should cooperate with each other, and that there was not a need to create an entirely new process because of the process. So let's focus on the product. If it's a product that would end up in FDA, they would have jurisdiction, USDA. Now, that's always challenging because you still at some point have to address how you made it. So there is a process component, but what the US has attempted to have as a longstanding framework is a product versus a process framing. That's not true with the convention and in other areas. And so there are just different framings, and they go back to risk perceptions or statutory components. And that's in part why risk benefit sometimes moves in and out.
Right. Okay, great. Thanks, Steve. One other question. Products with, quote, green credentials are increasingly being sought by consumers. What type of ag products might be optimum for us to develop using engineering biology, the tool of engineering biology? Question is, should we avoid food uses and focus on fiber materials to not get bogged down in regulation? I have an opinion on this, and as the moderator, I can't answer this question, but I'm going to leave it for you.
Well, I'll tee up something and then see if you take a whack at it, right? I think the if we accept for the moment the framework that I put, that it's actually a lab to environment transition, then the use of the material actually is not the big driver in whether or not there may be a regulatory challenge. Obviously, when you eat something, it is different than when you don't eat something, and if it's going to feed versus human consumption, all of those play into it. But the big hurdle for technologies has historically been getting across that laboratory threshold. Now, other areas, one of the reasons that the recombinant microbes became so difficult to move forward, whether it was in a remediation aspect or an agricultural aspect, or even in some cases, a national security or military aspect, right? There have long been fascinations of using microbes that would express GFP to report out hazardous materials, whether they would be explosives or other materials. So lots of fascination in those uses, but again, it's getting them out in the threshold or across that threshold. And there was the concern that you couldn't recall a living entity after you did it. That was part of the thing that Mycogen did. We took a dead recombinant out that made a Bt protein. And so you don't have to worry as much about recalling a dead organism as you would the ability to reproduce. That's where the macroscopic world has had an advantage with the crops, but not all crops. Again, obviously, there are a number of crops where they've been able to move into the environment and into the marketplace, and not for others, where you have concern about weedy relatives and other things of that framework. All of those are legitimate concerns. They need to be worked with. So then as you may think about a plant that would be used for a highly engineered plant that would use for some fiber production, you would have similar concerns. And that's, I think, the important point. Make sure that you don't do something in your technology development that stymied or slowed down the movement of some of those other plants that was really independent of the actual use. It was something fundamental with the biology or the techniques that were used within it. So certain kinds of promoters, certain kinds of other genetic elements. Think through that and try to do that effectively. I think that just as bio can mean lots of things, green can mean lots of things. And so I think it would be important if you're going to push that. I think the challenge is developing that narrative early, work with groups that may be impacted by that, and try to co-develop something instead of trying to view society or certain groups as adversarial. Figure out a way to come together and do as much as possible to try to identify benefits, legitimate concerns, move them forward.
Yeah, I think the comment that you used, and I like the phrase, the threshold from lab to environment, because the one thing that I think it's important for everybody on the call to realize is that there are ingredients that are already produced from recombinant microorganisms or microorganisms that are in our food system, like the heme proteins that are in Impossible Burger. They're not, the yeast has not been released into the environment. It's in a fermentation vessel controlled manufacturing process. Similarly with, rennin in the making of cheese is also derived from those systems, but it's that threshold of live release into the environment is where the hurdle is, not the end use application, is one comment I'd like to make. I also think that your comment, Steve, and re-emphasize the need to you need to build a big tent, and you need to have the comment you made about promoters, and you and I have talked about this a lot in the time that the 21 years I've known you, about the need to think about the design of your product from the beginning, so that you don't inadvertently, it's not a game of, you don't get caught in a corner. And again, bringing people along, connecting, building bridges, getting that conversation early and often, I think, is absolutely critical for any innovation, and particularly acute in the ag and food sector, about getting input and engagement up and down the value chain and across all the stakeholders, because we have the challenge in front of us of how do we produce more food environmentally sustainably, and how do we how can ag and food continue to be, it's one of the only industries that's large enough to be able to capitalize on, nature's preferred carbon sequestration method, which is photosynthesis, to be able to help mitigate climate change. So how do we use to add, we've heard about green and bio, I'm going to add the word natural into this in terms of helping address it. We have another question. How are recombinant enzymes now used in bakery and fermentation? Do you think food labels should clarify this and include this information?
So that's a great question. It's also back to a statutory question. And in part, because certain statutes exist that talk about the difference between something that is, I'll call it for the moment, this isn't the correct term, but a key component of the material that's being made versus a manufacturing aid. And so in many cases, the enzymes fall under this concept of a manufacturing aid or other terminology that is relevant for the domain. And so from a statute perspective, they may not end up needing to be regulated or labeled from that perspective. I think there are two anecdotes that are out there. I can't vouch that they're true. I think they're interesting anecdotes, but a lot of the vitamins that are produced that go into multivitamins are made by organisms that have had a recombinant past. And so when a large cereal producer was attempting to remove all GMO products from their framework, they actually had a difficult time finding materials to make some of the vitamins. And so a scientist who is at a university in the US and did a stint with the USDA actually kind of tracked this down and appears to have confirmed that the vitamin content of those particular materials had to shift because they couldn't get those components. Again, in certain states that in the US that were moving towards food regulation and packaging, certain enzymes used in the cheese arena, they did not have non-recombinant options at the time. And so there was some interesting stories about how that played out. Again, these may be anecdotes. They're kind of fun to read about, but the net being, it still comes down in many cases to a framing of what is statutorily required in any particular area.
Agreed. And I just think that it's interesting that we want to label, potentially concerned about labeling if it's derived from an engineered strain versus what's the natural source for some of these things. Well, it's the forced stomach of a calf. And that transparency is not there. in the context of the discussion. So again, I think it's the importance of the overall picture, understanding and helping build. And again, that's one of the things that we see as part of, and our next speaker in the series is going to be Jordan Thompson from Ontario Genomics. And Jordan is he's a key member of the Engineering Biology Steering Committee, the Canadian Engineering Biology Steering Committee, that's led by Bettina Hamlin from Ontario Genomics. And the regulatory component and framework is a key element to the roadmap, because we've seen where science and technology get ahead of the regulatory framework, and making sure that we have that level of engagement in the discussion from the beginning, so that we have a path to market, because that's the only way we make a difference, is being able to bring ideas into the marketplace so that they get used, and we can reduce and improve the efficiency at which we produce not just food, but also other materials, like you mentioned, new drugs, new biomaterials. Again, this is a disruptive technology, and it's imperative that we engage in the discussion with all stakeholders up and down the value chain. Thanks, Steve, for presenting today, and I really do appreciate, on behalf of the Global Institute for Food Security's Engineering Biology webinar series, thank you so much for presenting. And folks, thanks for being on the call, and we will have an opportunity to post this video along with the first one on the events page, and we will get back to answering it. We'll also provide answers to the questions that we addressed today. Thanks again, Steve.
All right,
Take care,
my
everybody.
pleasure.